Hi everyone. The lesson today is what is a skeletal muscle, and we'll start by reviewing the gross anatomy of skeletal muscle, and then we'll zoom into the micro anatomy closer and closer and closer until we get to the structure of the basic unit of a muscle fiber, which is the sarcomere. By the end of this lecture, you should be able to answer this question using most of this vocabulary, hopefully all of it. So just to give you a overview in GIFs. This is a cross-section of the spinal cord, so it represents signals coming from the brain in blue. And so you have these signals that are activating the entire muscle cell to contract. And it does that by basically triggering all of these muscle fibers that you're going to see when you cut open the muscle. It triggers all of these muscle fibers to contract together as one unit. So here's what that looks like in closer detail. Here you have one muscle fiber. Here you have a nerve ending with signals from the brain traveling through the entire muscle fiber. And these signals basically trigger these little yellow molecules called neurotransmitters to spread through the entire muscle cell and cause all of it to react at once. And we're going to talk more about this process in the next lecture in this series. Here you have a basic unit of a muscle fiber. It's called the sarcomere. And you have 1, 2, and a half, 2.5 sarcomeres pictured in this one window. So let's just review before we move on. What is the gross structure or gross anatomy of a skeletal muscle? So remember, this is the whole muscle right here. It's the muscle belly. And then here you have a fascicle. And you zoom in, you'll see a muscle fiber. So this is a single muscle cell right here. The muscle fiber is made of myofibrils. And if you zoom in some more, you'll see these thin strings called myofilaments coming out of the myofibrils. Why don't you pause the video, see if you can identify the levels of hierarchy of skeletal muscle structure, and when you're ready, you can resume. Okay, so once again, here's the muscle bundle. It's the whole muscle. It's made of fascicles, which are bundles of muscle fibers, and once again, these are single muscle cells. Muscle fibers are made of myofibrils, and if you zoom in some more, you'll see myofibrils are made of these protein strings called myofilaments. So if you want to go down this flow chart from largest to smallest, it's muscle, fascicle, muscle fiber, myofibril, myofilament. Okay, so now we're going to get into new content. Let's zoom in on one single skeletal muscle and how it's structured. So this whole thing is showing you one muscle cell, and the first thing you should notice is that it's multinucleate. Multi meaning many, nucleate meaning nucleus. That's because during fetal development, you'll have many muscle cells that fuse together to form one elongated muscle cell that has many nuclei so that the whole thing can react to a signal from your nerves all at once. So here you'll notice we have 1, 2, 3 nuclei. The other thing I want you to notice, besides the fact that you have multiple nuclei, which is really weird, is that in a typical cell in your body, you would expect the nucleus to be in the center of the cell. But in a muscle cell, they're pushed up right underneath the sarcolemma. And the sarcolemma is the plasma membrane that surrounds the entire muscle cell, but it has a special name because it has a specialized function. Right underneath the sarcolemma, you'll find the sarcoplasmic reticulum, which is not pictured here. I'll point it out in a future slide. But the sarcoplasmic reticulum is basically like your smooth endoplasmic reticulum, but it's specialized in the case of muscle cells to store calcium. So you have the nucleus and the endoplasmic reticulum or the sarcoplasmic reticulum pushed up right underneath this sarcolemma, this plasma membrane. Why is that? Why aren't they in the center of the cell? Well, that's because the entire muscle cell is full of these myofibrils that fill up pretty much the entire cytoplasm. And remember, it's because the function of the muscle is to contract all at once as one single unit in one fluid motion. And the way it does that is by ensuring that all of these myofibrils are coordinated. And the more myofibrils you have, by the way, the more power you have in the muscle contraction. So here you can see when the muscle contracts, the whole thing shortens all at once. And it only does that in response to signals from a nerve, which once again was shown in blue. By the way, that whole thing is called a motor unit, and we'll use that word again in the next lecture. So motor unit is one single nerve ending, shown in blue. Right, so this is one nerve, and here's one single nerve ending, and all of the muscle fibers in that entire bundle, all of the muscle fibers that respond to that one nerve ending, the whole thing is called a motor unit. So here is one muscle fiber in this motor unit. Here is one terminal to the nerve ending. And when a signal comes down the nerve ending, it's going to trigger those little yellow molecules to be released that then spread throughout the entire unit, like the entire muscle fiber, and trigger calcium to be released from the sarcoplasmic reticulum, which is underneath the sarcolemma, triggering all of these myofibrils to contract all at once. So how do the myofibrils contract? Here we have just one myofibril. So this would be like one organelle in the entire muscle fiber. And it's made of chains of tiny contractile units called sarcomeres. A sarcomere is found between two Z discs. So I want you to just take a second and identify where does the sarcomere, where are the boundaries of the sarcomere? Okay, here they are. So you have a Z disc here, you have a Z disc here, and this whole unit is called a sarcomere, and you have multiple sarcomeres along the entire length of the myofibril within the muscle cell. So the sarcomere is made of multiple, there are multiple sarcomeres in a myofibril, and each sarcomere is made of two types of myofilaments, actin and myosin. Here is the myosin shown in red, and here is the actin shown in blue. Once again, here's the myosin shown in red, and here's the actin shown in blue. And remember that they're bound by the Z discs, which form the boundaries of one sarcomere. Okay, so when we talk about the thick filaments, also known as myosin filaments, they're composed of a protein called myosin, and what's significant about them is that the myosin, the thick filaments, use ATP, which is a form of energy in the cell, and they use that energy to slide along the actin filaments to cause muscles to contract. The thin filaments, shown here in blue, are made of a protein called actin, but the myosin is what does the work of the muscle contraction, and we'll talk about how in the next lecture in this series. One thing to keep in mind is that when you look at the myosin, it has these little projections coming off of them. They look kind of like oar heads or golf heads, and what they do is they grab onto the myosin. Here you can see the actin, my bad. They grab onto the actin, which is shown in blue, and they pull themselves along the actin. And I'll show you an animation of that in the next lecture. You should also keep in mind that the myosin and the actin do overlap. And so here you can see that they're aligned so that if you look at this under the microscope, you would have actin and myosin in the same region over here and again over here. So there is major overlap between these two proteins, and there's an important reason for that. Okay, so once again, let's zoom in on the sarcomere. This is a single myofibril in a muscle fiber. So this is one organelle. And if you look inside of a sarcomere, you would see blue actin filaments and pink myosin filaments. And actin and myosin are called myofilaments. Let's just recap before we get into the nitty-gritty of the structure of a sarcomere. So you have a muscle fiber, that's the cell, that's made of myofibrils. Myofibrils are basically organized into multiple sarcomeres that have the myofilaments actin and myosin that pull against each other to cause the muscle to contract. Here's the muscle. Here is one fascicle in the muscle. Peel away one muscle fiber from this fascicle, and you'll see that this is what it looks like. This is one muscle fiber. This is the sarcolemma. It's the plasma membrane of the muscle fiber. Underneath, you have this web of yellow. That would be the sarcoplasmic reticulum. That's the endoplasmic reticulum of the muscle cell, and it stores calcium and releases it when the muscle needs to contract. The muscle fiber is made of these myofibrils, and if you cut away part of the myofibril, you'll see that it's got these lines on the inside, these myofilaments, actin and myosin, organized into block units called sarcomeres. Okay, so let's get into how a sarcomere is organized. The alignment of myofibrils and sarcomeres create banding patterns. So you have the I band shown here, where you have no myosin. Remember, the myosin are these red, thick filaments, and the actin are the skinny blue filaments. But there are places where there's no myosin, and those are called the I bands. And then, of course, there are places where there is myosin. Those are called the A bands. If you look at an A band, or if you look at a myofilament, myofibril, under a microscope, you would see that it would have a dark region where the myosin is, and it would have a light region where the myosin isn't. And so that dark and light banding pattern is characteristic of skeletal muscle fibers. The boundaries where the sarcomeres meet, they form this zigzag pattern where you have these scaffolding proteins that anchor the actin together, or anchor the actin to where they're supposed to be. And so that zigzag pattern is called a Z-disc. Z obviously for a zigzag. Right here, you have this M line, which is this region right in the very center of a sarcomere. So remember, from here to here is one sarcomere. In the very center is the M line, and then the H zone is this region where it's in the center of the sarcomere, and this is where you'll find no actin, just myosin. And it's called the bare zone because it's bare of actin filaments. So the reason why the H zone is significant is because when the entire sarcomere is relaxed, the Z discs will be farther away from each other, and the H zone will be larger. That's because there will be less overlap between the actin filaments and the myosin filaments. But when the muscle is contracted, the H zone becomes smaller. That's because the myosin filaments basically pull the actin towards the M line. And as the actin gets pulled towards the center, it causes greater overlap between the actin and the myosin, which shrinks the H zone. And so that's one of the ways that you'll know if you're looking through a microscope whether the muscle tissue is contracted or whether it's relaxed. If you see a large H zone under the microscope, it's relaxed. If you see a shrunken H zone, it's contracted. So here are your key takeaways when it comes to skeletal muscle structure. You should remember that skeletal muscles are bundles of muscle fibers. You should remember that muscle fibers, which are single muscle cells, are bundles of myofibrils, and that myofibrils are made of myofilaments. Remember that the myofilaments and the myofibrils are organized into block units called sarcomeres, and that's where the magic happens. So the actin and the myosin, which are the two myofilaments, are aligned in sarcomeres in a way that makes them have light and dark bands that are visible under a microscope. The light bands are where you have just actin, and the dark bands are where you have just myosin. I'm sorry, the dark bands are where you have actin and myosin. And the banding pattern changes shape during sarcomere contraction, where the H zone shrinks when the sarcomere is contracted, and it expands when the sarcomere is relaxed. Okay, you guys, let's see if you can go back to the essential question at the beginning of this lecture, and if you can answer the essential question using all or most of the vocabulary shown here, then that means that you have probably mastered this topic.